So with that, we can probably get started with Dr. Brian Roth from MSU.
Thanks for everybody to be here for being here. I'm just gonna present a quick overview of our efforts to characterize predator diets in Lakes Michigan and Huron over the past few years. My co-author list is very, very long. That's because this is a really intensive collaborative effort among multiple agencies, funding agencies, etc. In addition, there's a number of people that are important to thank, including creel agents, sea grant organizations, as well as volunteer anglers and sport fishing groups. And I'd like to do a special shout out to the U.S. Fish and Wildlife technicians who are out there collecting a lot of stomachs and stable isotope samples for us. So just to give a really quick overview, our project to evaluate these diets had a few specific goals. So for one, we wanted to determine species-specific diets for salmonine predators in Lake Michigan and Huron, also walleye. You'll see them in there here as well. And we want to do this lake-wide, but our sampling strategy was to survey Lake Huron and Lake Michigan as broadly as we could, both through time and across space. And so we wanted to determine seasonal trends in diet composition, as well as spatial patterns in diet composition. I won't be able to get into too much depth regarding both of those, but I hope to highlight some of the things that we see. In addition, as we move forward, one of the primary questions that always seems to come up again and again is how do what we see in the diets correspond to what, say, USGS collects in the prey fish surveys? And really, we wanted to be able to test that, and we're just kind of starting that process now. And so I'll highlight a couple of findings, at least preliminary findings. So just to give you an idea of what our sampling effort has been, it's been pretty substantial over the last several years. In 2015 and 2016, one of our collaborators collected stomachs as part of his master's thesis, but our group here at MSU has been collecting diets regularly since 2017, with a few exceptions. So 2020, of course, was COVID, where our ability to collect stomachs was relatively limited. And 2022, the project didn't have any funding. So we've also been collecting stomachs for the past 2 years as part of funding from the Great Lakes Fishery Commission. And we've done our best to try to increase our samples to pre-2022 levels. In 2024, we're still actually processing those stomachs, which is why the numbers are a little bit low. But overall, we have almost 30,000 total stomachs in our database. So this is both lakes. We access these in an access database, and certainly there are a few gaps in species and seasonally. So, for instance, species like brown trout and Atlantic salmon are relatively rare in the database. We just don't see many stomachs from them, and this is likely due some to angler effort, in that seasonally, in particular, we don't get many stomachs from like January or February, when there's probably not too many folks out on the lakes catching, say, Chinook salmon, um, as well as the ability to efficiently gather diets. So what I mean by that is, in those shoulder seasons or in the winter, anglers still are like going out on the ice if they're able to access it, or may go out for a day, but it's really difficult for us to cover both lakes simultaneously during that period because the anglers are dispersed over space. So I do want to give you guys an idea of how we actually get our data. So whenever we get a stomach, we visually identify its contents, and we identify fish to species. And with invertebrates that we find in our samples, we identify those to taxonomic order. So these would be like mayflies or dragonflies. We don't go down further than that. We measure and weigh the contents, and for fish, we're able to assess their prey length, and we back calculate these. A lot of the fish that we get in the stomachs are relatively digested. They look pretty gross. And so what we do is use a published formula for vertebrae and clythra, which I'll explain in a minute, and back calculate the length that the fish was when it was consumed. We also weigh each taxa, and I'll express diet proportions in terms of wet mass. So we see what's in the tray, and then we weigh it wet. Okay, we don't dry it or anything like that. So how can we tell fish species apart? Again, the stomachs are oftentimes full of pretty well digested fish. Here are the clytra, the pectoral fin supports. And each tax actually has a fairly unique shape of their clytra. And so we can oftentimes tell a species apart from clytra. Sometimes even those are missing. And in that case, we try to use their otoliths, their ear bones. So here's the difference between two pretty similar species. Here's deep water sculpin, which you can see are fairly arrow shaped, and mottled sculpin, which have this pretty prominent hook at the end. We can also use caudal bones as well if needed. So let me get into the results. Most fish didn't eat things like coal, robins, and Coca-Cola wrappers. They tended to eat fish. And each of the graphs are going to have a very similar format. I'm going to use these three-letter acronyms. And so ALE is alewife, which you're all familiar with. RGB is round goby. Inverts, I will explain whether they're like these, which are terrestrial, or aquatic, such as zooplankton. SMT, smelt. Perigonines include bloater, but also in some cases they're very hard to identify and separate. These are one kind of group that we have difficulty, but usually it's bloater that we're seeing. Sculpins, other, include kind of other taxa fish, and then yellow perch are separated out. And I can't see if there's questions, so Dan, if you don't mind, you can, let me know if there's a question that I should answer.
Sure, none so far, but I figured we'd put those in the Q&A box for those who are using the chat function. The Q&A box works well for the questions.
Great, thank you so much. So just to give you an idea of what those lake-wide diets look like, here's Lake Michigan, and you can see the 5 primary species that we capture out there, and there's a lot of light blue, which is alewife. And so not surprisingly, most taxa are in fact eating alewife. Chinook and coho in particular eat upwards of 97 to 99% alewife. A few taxa are a little bit different. Lake trout and steelhead or rainbow trout. Lake trout, like gobies, kinda tends to vary a little bit year for year, and although historically it maybe was increasing, particularly during the early part of this survey, it appears to have leveled off or even decreased since then. Steelhead, similarly, early in the data set appeared to like this light green, which are terrestrial invertebrates, but that has leveled off as well, and they're primarily just focusing on alewife now as well. Lake Huron is a completely different story. Not surprisingly, there was a collapse in alewife in the early 2000s, and so the predator species there tend to have to make different decisions about the foods that they're eating. And in fact, one of those results is our pretty diverse diets, and the proportions of those diverse prey items is pretty consistent. So, for instance, walleye tend to eat a number of yellow perch every other year, and when they're not doing that, they tend to eat gobies. Okay, steelhead. Remember, in the Lake Michigan, they're almost entirely eating alewife with some bugs thrown in. The opposite is true on Lake Huron, where they're primarily eating terrestrial invertebrates with a few rainbow smelt and maybe a few alewife thrown in there for good measure. Lake trout tend to really concentrate on 2 taxa, round goby, and they're in dark blue, and rainbow smelt. Coho tend to eat just a kind of random assortment of fish from year to year, but in recent years they've been really focusing on invertebrates. And Chinook salmon somehow are still finding alewife in Lake Huron. As it turns out, we only catch those Chinook salmon in the northern portion of the lake almost exclusively, and that is the only place where we see alewife in fish diets. So just to compare a couple individual species side by side, you can really see the difference between the 2 lakes here. So in Lake Huron, round goby are between 30 and 80% of the diet, depending on the year. but only 10 to 40% in Huron. So round gobies are much more important as a forage item in Huron than they are in Michigan. In addition, smelt is important as a pelagic prey fish in Lake Huron, whereas it's alewife, of course, in Lake Michigan. Chinook salmon, again, they're finding whatever alewife are there and eating them. But one of the things that kind of struck us about this is the amount of invertebrates. And in this case, a lot of those invertebrates that we're seeing in Chinook stomach diets are not terrestrial, but rather they're bathytrophs, they're zooplankton, which indicates they're not, there's no alewife. They're not finding, real good alternative invertebrate prey. And of course, it's exclusively alewife in Lake Michigan. Steelhead really represent a contrast as well. So they fed extensively on inverts. Upwards of 70 to 75% of their diets is terrestrial insects in Lake Huron. And again, if they have the opportunity to find alewife, they do. They eat them. So do these diets, really represent what's going on? Well, kind of, right? So when we look through time, we see some pretty consistent trends across years. Now, all the years have been combined into this graph, but every single year we see a similar pattern. In particular, lake trout, brown trout, and steelhead in Lake Michigan tend to transition away from some other prey to alewife as the year goes on. In this case, the month 4 would be April and 10 would be October. So in springtime, they tend to concentrate on some other prey. For lake trout, it's round goby. For steelhead, it's those invertebrates. And for brown trout, it's this kind of mix of different prey items. And then they all transition to alewife as the summer goes on. So by August, they're all focusing really heavily on alewife. In Lake Huron, not surprisingly, it's a little bit more complex, but we do see some similar trends. So in springtime and early summer, for lake trout and walleye, we see them really focusing on gobies through June in this case. And then they'll transition to some other prey usually. So for lake trout, it tends to be rainbow smelt here. And for walleye, it tends to be yellow perch. And so there's this interesting transition from being benthically oriented foragers to those that are more pelagic as the year goes on.
And Brian, if I can interject, we do have a question.
Please.
Is Saginaw Bay included in these results?
Absolutely. Yep. So Saginaw Bay is included in these results. This is meant to be a relatively quick overview. However, if we were to divide out Saginaw Bay, you'd see some really interesting results. So walleye, for example, don't eat many round goby in Saginaw Bay proper. Rather, they eat some mix of inverts, cyprinids, random prey fish. So we've actually found baby Chinook in their diets in Saginaw Bay. And then as we go into summer, they'll really start to hammer yellow perch in Saginaw Bay. Outside the basin, they're almost exclusively eating round goby. All right, so I'll move into kind of the next component, which is, do prey fish surveys and predators agree? Again, this is a really common question that has come up. And what we're really after here is just to determine how well can those prey fish surveys actually inform us regarding what predators are eating. Now, again, we've just begun this analysis. And so what I'd like to do here is kind of compare what sizes of prey we're seeing in the diets versus what we see in the survey, just to see how much those match up. So for one, I'll focus on lake alewife consumption in Lake Michigan. And just to orient you to this figure, what you see here are what are called size distribution plots, where the length of the prey in the stomach is on the x-axis here, and kind of how abundant those prey, that size is in predator stomachs is on the y-axis. So a taller peak means that size corresponds to it was very abundant in the diets. And this is for every predator that we have. So each color represents a different predator. And one of the interesting kind of things that we see is that in some years, the predators are really focusing on a single length. So if you look at 2017 here, you see that all the predators were consuming alewife that were approximately 100 millimeters or so. In some years, they do tend to spread out a little bit, but they tend to kind of follow a pretty similar size of alewife. So they're kind of keying in on similar sizes. So now I've lumped all those predators into a single line here in blue, and then compared that to what we saw from the prey fish surveys and just graphed them together to give you an idea. And what we see is that there is quite a bit of overlap between what we see in the prey fish surveys versus what we see in predator diets, but not always, right? There's some really interesting differences here that are really worth investigating. So for instance, here in 2022, predators really focused in on this smaller size of alewife that was less than 100 millimeters, whereas the prey fish surveys found a lot of much larger alewife, which was interesting because in the following year, then that's what predators were focusing on. And so I think that as we go along, this sort of analysis offers to give us a preview into some really interesting predator-prey dynamics that we might not have observed before without this time series. I'll show you a similar graph for rainbow smelt on Lake Huron, where again, predators are consuming similar sizes of rainbow smelt. There's a little bit diversity. So in 2023, the 3 species that we found consuming smelt were kind of consuming 3 different peaks, right? But usually they overlap pretty well. And then when we do a similar analysis or investigation where we look to see how together as a community are they foraging on the same sizes that the surveys are finding, the answer is it's not as clear cut, right? And so there's kind of some really interesting things going on here that we're not sure. So for instance, in some years, again, we're pretty close. So in 2019 here, survey and predators were pretty close in terms of the sizes of smelt they're feeding on. But in 2023, again, the survey found lots and lots of small rainbow smelt and very few large ones. And the predators, that's what they were eating. And so there's the potential for a number of explanations of this, right? So, our surveys of predator diets were earlier in the year, and what the surveys captured is what's left over after the predators already had their fill. Right? That could explain something like this. But some of these patterns are just a little bit difficult to digest, and it's going to take some time and thought to be able to really make a clear picture out of it. Lastly, I'll get to some frequently asked questions. And one of those is, how much do lake trout eat salmon? That's one of the most common questions I get at workshops like this. And the answer is not much. Out of over 16,000 lake trout that we evaluated, we found a total of 96 salmon. And that's, 0.22% of all the prey that we've looked at. We find that more on Huron than in Lake Michigan, but as it turns out, coho ate more Chinook salmon than lake trout did on Lake Michigan. The last one is about bloater. So how much bloater do Chinook eat? And not surprisingly, more in Michigan than in Huron, but about 0.9% of all the prey that we've looked at in Chinook salmon are bloater. And we average about 10 bloater and stomachs per year out of about 450 Chinook salmon diets we looked at. But we have seen a slight uptick in 2024 so far, where we found 16 bloater and only 321 stomachs. So maybe their consumption of bloater is increasing. We're not sure. So to summarize, the diets are pretty stable. We see pretty similar diet compositions. There's a little up and down, but they're pretty stable across lakes. Lake trout, steelhead, and brown trout are certainly the most variable among years. I didn't have time to talk to you much about location, but there is quite a bit of variation in stomach contents by location, particularly for lake trout and brown trout, and through time, through the seasons. Huron diets are very diverse relative to Lake Michigan, And we still have a lot of work to do to determine how predation relates to those prey fish assessments. This project is winding down. We end in early spring 2026. So we're going to finish up our 2024 stomachs, collect a few more in 2025, and continue our analysis. Thank you. Any questions?
Let's see. We have one more from the Q&A box. Are the stomach contents saved and archived or disposed of after analysis?
Oh, they're definitely disposed of. Yeah, I mean, we freeze the stomachs, and people already complain to me about the smell as they walk by my lab. Not surprisingly, alewife do not smell good when they're partially digested. And so, yeah, we do collect as much data as we can, and then we just toss them out.
Thanks. Any more questions for Brian? If not, thanks a lot for joining us, Brian. Thanks everyone for attending.